Welcome to Chapter 21, Lipid Biosynthesis. In this lecture, we will be covering the first part of this chapter, that is, fatty acid biosynthesis. Learning goals for this chapter involves understanding the biosynthesis and regulation of a variety of lipid molecules. This involves fatty acids, eicosanoids, triacylglycerols, membrane lipids, and last but not least, cholesterol, which is a form of sterol. Now, it is important that you recollect all your organic chemistry knowledge. What we are going to look at in this chapter is the biosynthetic route, which involves chemical mechanisms catalyzed by enzymes. So, it is important that we merge chemistry and biology knowledge together to understand the Lipid Biosynthesis chapter. Lipids fulfill a variety of biological functions. Some of the common biological functions of lipids are shown in this slide. In addition, a variety of cellular roles of some of the lipids are recently recognized. There are many different functions of lipids that are still not known. The ability to synthesize a variety of lipids is essential to all organisms. This chapter will describe the biosynthetic pathways for some of the most common cellular lipids and the regulation of these lipids in cells. To begin with, let us look at the biosynthesis of fatty acids. Now, you may think that biosynthesis of fatty acids is essentially the reverse of beta oxidation, but that is not the case. Now, the metabolism of fatty acids involves two parts: catabolism and anabolism. Catabolism of fatty acid is what produces energy. Now, we have seen that catabolism produces acetyl-CoA through beta oxidation. It also produces NADH and FADH2, right? The reducing equivalents of ATP. And this happens in mitochondria. Whereas anabolism of fatty acids require energy. This means that it requires acetyl-CoA and Malonyl-CoA, which is the starting material for fatty acid biosynthesis. It also requires the reducing power from NADPH, and it takes place in cytosol in animals and in chloroplast in plants. Next, let's look at the subcellular localization of lipid metabolism and compare it in animal cells and plant cells. We know that mitochondria in animal cells is where fatty acid oxidation takes place. It is also where acetyl-CoA and ketone body synthesis happens. In addition, fatty acid elongation also takes place in mitochondria. Whereas in plant cells, peroxisomes is where fatty acid oxidation takes place. Now, where does fatty acid biosynthesis happen? This happened in the cytosol in animal cells. Now, cytosol is where the concentration of NADPH is high, and that is primarily the reason why fatty acid biosynthesis takes place in cytosol. Whereas in plant cells, chloroplast is where fatty acid biosynthesis happens. In addition to this, other biosynthesis such as phospholipid biosynthesis and sterol biosynthesis takes place in the endoplasmic reticulum of animals and plant cells. It is also where fatty acid desaturation happens. Fatty acid synthesis occurs in cell compartments where NADPH levels are high. But what are the sources of NADPH? In animal cells, and to be specific, in adipocytes, the pentose phosphate pathway and the malic enzyme pathway are the sources of NADPH. Whereas in hepatocytes and mammary glands, pentose phosphate pathway is primarily the source of NADPH. Photosynthesis is the source of NADPH in plants. These are the two pathways where NADPH is produced. primarily in animal cells. The first one is the conversion of malate to pyruvate that is catalyzed by malic enzyme. In this process, it converts NADP to NADPH. The second pathway is the pentose phosphate pathway. If you recollect your basic biochemistry course, glucose 6-phosphate gets converted to ribulose 5-phosphate via the action of different enzymes, and in this case 2-NADPH is released. So pentose phosphate pathway is a major source of NADPH. As I said before, the biosynthesis and breakdown of fatty acids occur by different pathways and are catalyzed by different sets of enzymes and also take place in different parts of the cell. So, fatty acids are built in several passes, processing one acetate unit at a time. The acetate unit comes from an activated malonate in the form of Malonyl-CoA that is shown here. Each pass involves reduction of a carbonyl carbon to a methylene carbon. Now, what does that mean? Before we get into the details of fatty acid biosynthesis, let's look at how Malonyl-CoA is formed from Acetyl-CoA. The formation of Malonyl-CoA is catalyzed by an enzyme, Acetyl-CoA carboxylase, and this involves a carboxylation reaction. This enzyme has 3 subunits. One of this subunit has biotin covalently linked to a lysine residue. Remember the reaction catalyzed by carboxylases? If you recollect the previous chapter, you would remember it. Biotin is a cofactor that carries CO2 in the form of bicarbonate. In animals, all 3 subunits in Acetyl-CoA carboxylase are one polypeptide chain. Also, bicarbonate is the soluble source of carbon dioxide. Acetyl-CoA carboxylase catalyzes a 2-step reaction that is similar to carboxylation catalyzed by pyruvate carboxylase and propionyl-CoA carboxylase. We have seen both these enzymes before. Carbon dioxide binds to biotin. This is the structure of biotin. Right? Now, biotin is linked covalently to a lysine side chain to a biotin carrier protein, and that is one of the subunit of Acetyl-CoA carboxylase. Shown here is the structure. This is a cartoon representation for Acetyl-CoA carboxylase. The 3 subunits are shown here. Biotin carrier protein is one of them. Biotin carboxylase is another subunit, and a trans-carboxylase subunit is where this carboxylate is transferred to Acetyl-CoA. In the first step, carboxylation of the biotin group happens in the biotin carboxylase subunit. In the second step, the enzyme undergoes a conformational change to carry this carbamoyl functional group to the trans-carboxylase subunit. As you could see, there's shifting this specific group to the trans-carboxylase subunit. And this is where acetyl-CoA binds. And once it binds, this carboxyl group is transferred to this carbon. And as a result, acetyl-CoA gets converted to Malonyl-CoA. Fatty acid synthesis is catalyzed by the enzyme fatty acid synthase, or FAS. FAS catalyzes a repeating 4-step sequence that elongates the fatty acyl chain by 2 carbons at each step. It uses NADPH as the electron donor. In addition, it also uses 2 enzyme-bound thiol groups as activating groups. There are 2 major variants of fatty acid synthesis. Fatty acid synthase 1, or FAS1, is found in vertebrates and fungi, and fatty acid synthase 2, or FAS2, is found in plants and bacteria. The FAS1 found in vertebrate consists of a single multifunctional polypeptide chain. The mammalian FAS1 is a prototype of this. Phase 1 systems leads to a single product during fatty acid biosynthesis, and no intermediates are released. We'll talk about the phase 1 system in detail. A crystal structure of the mammalian fatty acid synthase is shown here. This protein consists of a single multifunctional polypeptide chain, and it functions as a homodimer. There are 7 active sites for different reactions, and all these active sites lie in separate domains. The different enzymatic active sites are beta-keto acyl-ACP synthase, or KS, Malonyl-Acetyl-CoA ACP transferase, or MAT, beta-hydroxyAcyl-ACP dehydratase, or DH, enoyl-ACP reductase, or ER, and beta-keto-acyl-ACP reductase, or KR. ACP is the acyl carrier protein. We'll talk about this in the upcoming slides. Now, there's another domain called the transesterase or the thioesterase domain that releases the final palmitate product from the ACP when synthesis is complete. On the right here is a schematic representation of this mammalian fatty acid synthase protein, and all the enzymes in this specific protein are shown, and these are the enzymes that catalyzes the 4 major steps of the fatty acid biosynthesis. Although different, the beta oxidation pathway is similar to the fatty acid biosynthetic pathway in at least one aspect. If you recollect, the beta oxidation pathway involves removal of 2 carbons from the fatty Acyl-CoA chain in one single step. In the same way, the fatty acid biosynthesis involves attachment of acetate unit, which is 2 carbon unit, from Malonyl-CoA to a growing chain, and then reducing it. So 2 carbons are added at one time. The reaction involves cycles of 4 enzyme catalyzed steps. The 4 reactions are condensation of the growing chain with activated acetate, reduction of a carbonyl to a hydroxyl group, dehydration of an alcohol to a transalkene group, and reduction of this alkene to an alkane. The growing chain is initially attached to the enzyme via a thioester linkage. The acyl carrier protein serves as a shuttle in the fatty acid biosynthesis. Like I said before, ACP, or the acyl carrier protein, is an important part of the fatty acid synthase unit. This protein contains a covalently attached prosthetic group, and the structure is shown on the right here. And it has this 4-phosphopantathene group, which contains a pantothenic acid moiety in this, and this acts as a flexible arm to tether acyl chain while carrying intermediates from one enzyme subunit to the next. The way it tethers the acyl chain is by using the thiol moiety, that is the SH group, which esterifies malonyl groups. This delivers acetate in the first step, or malonate in all the subsequent steps to the fatty acid synthase. Shuttles the growing chain from one active site to another during the 4-step reaction. This slide describes the sequence of the steps involved in the fatty acid biosynthesis. In the first step, a malonyl group and an acetyl group are attached to the thiol in fatty acid synthase. Condensation results in the release of carbon dioxide, and the next step involves reduction reducing a carbonyl group to a hydroxyl group. The third step involves a dehydration, where this hydroxyl group is removed to to form an alkene. And this alkene is further reduced by NADPH to form an alkane. So this is the first sequence of the steps involved in fatty acid biosynthesis. Let us look in detail the reactions catalyzed by the mammalian fatty acid synthase. Now, in this slide, the charging and activation of the acyl carrier protein is described. As I said before, there are two thiols in the mammalian fatty acid synthase. One of the thiols is in the acyl carrier protein ACP, and the other thiol is in the KS domain, or the beta-keto acyl-ACP synthase domain. Now, these two thiols must be charged with the correct acyl groups before the condensation reaction, or before the first step of fatty acid biosynthesis can begin. The acetyl group of the Acetyl-CoA that is shown here is first transferred to the acyl carrier protein, and this is catalyzed by the enzyme Malonyl-CoA ACP transferase. Once this is done, the ACP passes this acetate to the cysteine of the beta-keto acyl-ACP synthase, which means the thiol goes from here to here. That is shown in this slide. It has transferred this to this thiol. So it's a trans-esterification reaction. Once this is done, the ACP is then recharged with Malonyl-CoA, and after this you will have the enzyme that has two thiols attached to proper acetyl groups, and this is ready for the first step of fatty acid biosynthesis. The first step involves the formation of a beta-keto intermediate. This is a formal Claisen condensation reaction, and in this reaction the carbonyl carbon of the acetyl-CoA molecule is attached by this carbon, and as a result carbon dioxide is released, and this results in the formation of a beta-keto ester. Now, this creates a beta-keto intermediate, and it's catalyzed by beta-keto acyl-ACP synthase. And this coupling reaction, this coupling of the condensation to the decarboxylation reaction of Malonyl-CoA makes this reaction energetically favorable. Now, as you could see, the mechanism of this reaction is pretty straightforward. Electrons are pushed into this carboxyl, and as a result, the bond, which is electron rich, attacks the carbonyl carbon of acetyl-CoA, and as a result, this thiol from the KS, or the beta-keto acyl-ACP synthase, is relieved, and now the product is stuck to the ACP domain. Remember what I said before, ACP has this tether, and this is what carries the substrate or the product around for various different reactions. Once this beta-keto acyl intermediate is formed, it is then taken to the next enzyme for the next reaction. Step 2 involves formation of B-hydroxybutyryl-ACP, as shown here. This is a reduction reaction, and in this reaction NADPH is reduced to form NAD+. The enzyme involved is beta-keto acyl-ACP reductase, or KR. So ACP brings the product near this specific enzyme, and it uses NADPH to reduce this carbonyl to an alcohol. So the specific carbonyl is shown and is shaded in yellow here. The product beta-hydroxybutyryl-ACP is formed. This is then carried to the next enzyme by ACP for further processing. Step 3 involves formation of trans-delta-2 butynyl ACP. This is basically a dehydration reaction, which means that removal of a hydroxy group and
a hydrogen to form an alkene intermediate. And as I said, it creates a trans double bond. The OH and a hydrogen is removed from C2 and C3 of beta-hydroxybutyryl-ACP. This is catalyzed by beta-hydroxy-acyl-ACP dehydratase. A short mechanism of this reaction is shown on the right in this red colored box. This hydrogen, which is acidic because it's next to a carbonyl carbon, is abstracted by a base, and this pushes the bond inside and pushes out hydroxy group, which takes up a proton and gets expelled as water. As a result, a trans double bond is formed, and this is exactly the same structure as shown here. Step 4 involves the formation of Butyryl-ACP. So this trans delta 2 butynyl ACP is now reduced to an alkane. That's the reaction that happens here. This is catalyzed by enoyl ACP reductase. And this enzyme uses NADPH as a cofactor, which donates electron to reduce the double bond. And as you could see, the ACP or the acyl-CoA protein has now carried the product to ER or enoyl ACP reductase, which reduces it to the alkane. And this is now again brought back near KS. Once Butyryl-ACP is formed, it is then translocated near the beta-keto Acyl-ACP synthase domain for a trans-esterification reaction. This means that the butyryl group from the thiol of ACP is transferred to the thiol of the beta-keto Acyl-ACP synthase. So this is the result of that. Now, this is the final step of the first round of fatty acid biosynthesis, which means that starting from an acetyl group attached to the thiol of the beta-keto Acyl-ACP synthase, you end up with a butyryl group. So 2 carbons are added to the acetyl groups after the first round of fatty acid biosynthesis. From here on, this intermediate is taken for further addition of 2 more carbon atoms. So this is catalyzed again by malonyl acetyl transferase. This intermediate at this stage, a Malonyl-CoA is transferred, a malonyl group is transferred from Malonyl-CoA to the ACP thiol. And as a result, you end up with this intermediate, and this initiates the first step of the next round of fatty acid biosynthesis, that is condensation. And as a result, you end up with another intermediate with 2 more carbon atoms added. The fatty acid synthase reactions are repeated to form the 16-carbon palmitate. The fatty acyl chain grows by 2 carbon units donated by activated malonate, with the loss of carbon dioxide at each step. The initial acetyl group is shaded in yellow. The C1 and C2 of malonate group is shaded in orange, and the carbon released as carbon dioxide is shaded in green. After each 2 carbon addition, reductions convert the growing chain to a saturated fatty acid of 4, then 6, then 8 carbons, and so on. The final product is thus a 16-carbon palmitic acid. As I said before, there are 6 enzyme subunits in the fatty acid synthase protein, and these carry out various different reactions. For example, beta-keto Acyl-ACP synthase carries out the condensation reaction with acetate. The beta-keto Acyl-ACP reductase enzyme carries out the reduction reaction, the first reduction reactions, where it reduces a carbonyl functional group to a hydroxy functional group. A dehydration of alcohol to alkene is catalyzed by beta-hydroxy-acyl-ACP dehydratase. A second reduction reaction of a transalkene to an alkane is catalyzed by enoyl-ACP reductase. The Malonyl-Acetyl-CoA ACP transferase is involved in chain transfer and in charging new malonyl groups to the ACP protein. A final enzyme known as thioesterase is involved in the final step of fatty acid biosynthesis. What it does is it cleaves the palmitate from the ACP chain, and that's how palmitate is released. 7 cycles of condensation and reduction produce the 16-carbon saturated palmitoyl group that is still bound to ACP. For reasons not well understood, chain elongation by the synthase complex generally stops at this point, which means that once palmitate is formed, the chain elongation stops. And at this point, thioesterase, the enzyme that I talked about in the previous slide, hydrolyzes this palmitate from ACP. So the thioester is hydrolyzed. So we can consider the overall reaction for the synthesis of palmitate from acetyl-CoA in 2 parts. First, the formation of 7 Malonyl-CoA molecules from acetyl-CoA. Remember, Malonyl-CoA is formed from acetyl-CoA. Since there are 7 steps, 7 acetyl-CoA gives rise to 7 Malonyl-CoA. In the second part, we take into account the condensation, reduction, hydration, and the second reduction using NADPH to produce a palmitate molecule. So, if you balance the equation, 8 acetyl-CoA molecules in the presence of 7 ATP and 14 NADPH gives rise to a palmitate molecule and 7 ADP along with 8 CoA and 14 NADP plus molecule. This also produces 6 water molecules. Notice that only 6 net water molecules are produced. This is because one of them is used to hydrolyze the thioester linking the palmitate product to the ACP domain of the fatty acid synthase. In eukaryotes, there is an additional cost to fatty acid synthesis because acetyl-CoA is generated in the mitochondria and must be transported to the cytosol, because cytosol is where fatty acid biosynthesis takes place. So, in order to do that, you need another 2 ATP molecules. So, transportation of acetyl-CoA into cytosol involves the cost of 2 additional ATP molecules. Therefore, cost of fatty acid biosynthesis is 3 ATPs per 2 carbon unit. 2 for transporting acetyl-CoA and 1 for converting acetyl-CoA to Malonyl-CoA. And this is for 1 2 carbon unit. In addition, remember that NADPH is being utilized. In eukaryotes, nearly all acetyl-CoA molecules used in the fatty acid biosynthesis is produced in the mitochondria. That comes from pyruvate oxidation and from catabolism of the carbon skeletons of amino acids. Remember that the acetyl-CoA arising from the beta oxidation of fatty acids is not a significant source of acetyl-CoA for fatty acid biosynthesis in animals, because the 2 pathways are reciprocally regulated. The inner mitochondrial membrane, as shown in this figure, is practically impermeable to acetyl-CoA. So, an indirect shuttle transfers acetyl group equivalents across the membrane. In the first step, acetyl-CoA from the source that I just described gets converted to citrate by citrate synthase. Citrate gets out of the inner mitochondrial membrane and out of mitochondria through citrate transporter. In the cytosol, the enzyme citrate lyase converts citrate to oxaloacetate. And in this process, one equivalent of ATP is used. And once this converts it to oxaloacetate, a molecule of acetyl-CoA is released. And this is the acetyl-CoA that is being used for fatty acid biosynthesis. Oxaloacetate that is produced on the cytosolic side of mitochondria cannot re-enter mitochondria because there is no transporter for oxaloacetate. And hence, an enzyme, malate dehydrogenase, converts oxaloacetate to malate, and in the process it uses an NADH. Malate can pass through a transporter and get into the mitochondria and gets converted to oxaloacetate by another malate dehydrogenase on the matrix of the mitochondria. But it so happens that a majority of malate is converted to pyruvate by the enzyme by the malic enzyme. The reason is that this conversion can result in NADPH. So the pyruvate that is formed, a majority of pyruvate is formed from malate, re-enters the mitochondria via pyruvate transporter. And once it enters the mitochondria, pyruvate is converted to oxaloacetate by pyruvate carboxylase. Remember, pyruvate carboxylase that is involved in the citric acid cycle. And in this process, another equivalent of ATP is used. So, the net result is that to convert or to transfer acetyl-CoA out of the mitochondria, 2 ATP molecules are used. When a cell has more than enough metabolic fuel to meet its energy needs, the excess is generally converted to fatty acids and stored as lipids such as triacylglycerols. The reaction catalyzed by acetyl-CoA carboxylase, that is, in converting acetyl-CoA to Malonyl-CoA, is the rate-limiting step in the biosynthesis of fatty acids. This enzyme, acetyl-CoA carboxylase, is an important site of regulation. In vertebrates, specifically, palmitoyl-CoA is the principal product of fatty acid biosynthesis. This product, that is palmitoyl-CoA, is also a feedback inhibitor of acetyl-CoA carboxylase, as you could see from this image here. Citrate, on the other hand, is an allosterically activator. As you can see, the green circle indicates that it's an activator. The red circle with a cross indicates that it's an inhibitor. Citrate plays a central role in diverting cellular metabolism from the consumption of metabolic fuel to the storage of fuel as fatty acids. When the concentrations of mitochondrial acetyl-CoA and ATP increase, citrate is transported out of mitochondria. It then becomes both the precursor of cytosolic acetyl-CoA and an allosteric signal for the activation of acetyl-CoA carboxylase. Acetyl-CoA carboxylase is also regulated by phosphorylation. Phosphorylation triggered by the hormones glucagon and epinephrine inactivates the enzyme and reduces its sensitivity to activation by citrate. As a result, acetyl-CoA is not converted to Malonyl-CoA, and Malonyl-CoA level drops. Palmitate, the principal product of the fatty acid synthase system in animals, is also a precursor of other long-chain fatty acids. Palmitate can be lengthened to longer chain fatty acids by the fatty acid elongation systems in the smooth endoplasmic reticulum and mitochondria to create longer fatty acids. As in palmitate synthesis, each step that is performed by the fatty acid elongation system also adds unit of 2 carbons. Stearate, which is an 18-carbon fatty acid, is the most common product of the fatty acid elongation system. Palmitate and stearate can also be desaturated. This means that the saturated fatty acids, palmitate and stearate, can be converted to monounsaturated fatty acids, palmito-oleate and oleate. This is catalyzed by the enzyme fatty Acyl-CoA desaturase. This enzyme requires NADPH to catalyze this reaction. It also uses cytochrome B5 and cytochrome B5 reductase to transfer electrons. Molecular oxygen is a key player in the reaction catalyzed by fatty Acyl-CoA desaturase. Conversion of a saturated fatty Acyl-CoA to a monounsaturated fatty Acyl-CoA is an oxidation reaction. And in this process, this enzyme uses NADPH as a cofactor. Now, in converting a saturated fatty Acyl-CoA to an unsaturated fatty Acyl-CoA, cytochrome B5 is also used. As you could see, the Fe2 plus in cytochrome B5 is converted to Fe3 plus. At the same time, to maintain the cycle, cytochrome B5 reductase uses FADH2 and reduces Fe3 plus back to Fe2 plus while converting FADH2 to FAD. NADPH then comes into picture and it reduces FAD back to FADH2 while getting oxidized to NAD+. In this process, oxygen accepts 4 electrons from 2 substrates, 2 electrons from the saturated fatty acid and 2 electrons that is coming from the ferrous state of cytochrome B5. It is important to remember that plants can desaturate positions beyond C9, but humans cannot. Humans have delta 4, delta 5, delta 6, and delta 9 desaturases, but cannot desaturate beyond delta 9. As a result, plant can produce linoleate, which is the 18:2 delta 9-12 polyunsaturated fatty acid. Humans cannot produce linoleate and has to get linoleate from diet. Linoleate is an essential polyunsaturated fatty acids for humans. And PUFAs or polyunsaturated fatty acids are precursors to eicosanoids